1991, hikers in the Alps stumbled on the body of a man, mummified and frozen. It was visible because the ice was melting. After looking over the body, the hikers realized that this wasn't the body of one of the hikers who go missing every year in this alpine region. This was a man who had died more than 5,000 years ago. In fact, after careful study, scientists found that he is the oldest human ever found, a man 5,300 years old. Scientists call him Ötzi, or the Iceman. How he died became clear pretty quickly. Inferring from the evidence, an arrowhead lodged in the Iceman's back led to the conclusion that he was murdered. Ötzi was an immediate archaeological sensation. Research teams eagerly studied both the mummy and his incredibly well-preserved clothing: leather shoes, a woven grass cloak, and goatskin leggings. From these remains, could archaeologists predict who he was, how he lived, and why he died up here in the mountains? Based on the evidence, many researchers thought that Ötzi must have been an alpine shepherd, leading goats and sheep to high-altitude pastures in the summer. Shepherds today still herd livestock along the same path where Ötzi was found. If Ötzi was a shepherd, perhaps he was killed for his sheep. Two anthropologists, Sylvia Reinhardt and Hans Plotzkammer, and Anna Pedrotti, an archaeologist, decided to test this theory. They started by making an exact duplicate of Ötzi's clothes. The clothes would help the researchers model how Ötzi lived. When they were done, Hans donned the outfit. He would find out firsthand whether it could have held up to the tough life of an alpine shepherd. As Hans set off up the mountain on foot, Sylvia and Anna took the easy route, by helicopter. For Hans, it was a grueling 1,200-meter climb to model the Iceman's apparent journey. From the mountaintop, Sylvia and Anna could see that Hans was having trouble. At last he arrived exhausted. His shoes were a disaster. They had no traction and they hadn't kept the water out at all. The poor performance of the shoes indicated that Ötzi probably wasn't a shepherd. Someone wearing shoes like this couldn't make the 1,200-meter mountain climb regularly. But if Ötzi wasn't a shepherd, why was he up here? And why was he killed? Years of examination have not yet come up with an answer, but researchers have not given up. They are confident that with more study, and maybe even new technology, the Iceman will reveal the story of his life and death. How did the scientists follow the steps of observing, inferring, predicting, and modeling in their study of the Iceman? As you watch this segment, think about how investigators collect, store, and analyze evidence from a crime scene, and what they are able to infer from observations, new data, and additional evidence. Forensic science has become a cornerstone of crime solving. It is almost impossible to commit a crime without leaving a trace. And forensic experts can use even the most minute of these clues to identify a criminal. But this was not always so. Before modern scientific advances developed, crime solvers relied on confession, eyewitness accounts, and the most obvious of clues. 200 years ago, John Toms had a business dispute with his neighbor. Enraged, he vowed to get revenge. Toms carefully loaded his pistol, first with gunpowder, then with a wad of paper torn from a song sheet to hold the gunpowder in place. He placed the rest of the song sheet in his breast pocket. Toms made his way to the neighbor's yard, where his target was strolling. He aimed, fired, and hit his mark. Then he approached his victim. While bending over, the wounded man grabbed hold of Tom's sleeve for a brief moment before he died. Toms quickly ran away. The sheriff came to investigate and quickly found the clues that led him to suspect Toms. He found on the victim the wad of paper Toms used to secure the gunpowder. When the sheriff's men questioned Toms, they found the rest of the song sheet just where he left it, in his pocket. Quickly, they were able to match this paper to the wad removed from the crime scene.
Take him away.
And Toms was arrested. He was tried, found guilty, and hanged for his crime. In the last 200 years, detectives have come to rely more and more on forensic evidence. Today, with modern scientific tools in hand, this investigation would be handled quite differently. A scientific method would be employed using observing, inferring, predicting, and modeling. Based on this information, scientists would develop a hypothesis about who the criminal was. Toms left a slew of clues that modern forensic experts could use to link into the crime. Even if John Toms had burned the song sheet and made efforts to disguise other clues, experts would still have plenty to go on. First, they would secure the crime scene to protect any evidence. They would also take photos of the entire scene. Then, forensic investigators would take the temperature of the victim to determine the time of death. This data would help scientists form a hypothesis about the identity of the criminal.
Body temperature 18 degrees C.
Detectives would systematically comb through the crime scene for other clues, collecting other data. A footprint that Toms left in his escape would be cast in plaster, which would show that Tom's boot matched perfectly, down to a chip on the heel. Continuing to collect data to support or refute the hypothesis, the forensic team would examine the body for hair or other evidence. They would look under fingernails for bits of fiber. Collected with adhesive tape, the fibers would be taken to the lab for analysis.
Can you remove your coat and put it in the bag?
The team would gather clothing from the suspect to compare to fibers found at the crime scene. The fibers in Tom's vest make a perfect match. Blood on the victim's hand would lead detectives to believe that they would also find the victim's blood on the suspect. Tom's blood-stained shirt would be collected and analyzed. In the lab, scientists would use DNA analysis to test the blood on the shirt and compare it to the victim's. Because each person has a unique DNA code, the results of this kind of test are generally conclusive. The blood on Tom's shirt would definitively match his victim's. The gunpowder Toms used would also provide clues. It leaves a residue on the hands and face of the shooter. Samples from the victim's wound and swabs from Tom's face would be analyzed in a crime lab. Using a technique called chromatography, which separates chemicals in the gunpowder, samples would show that there is a perfect match. Forensic scientists would now form a conclusion based on the data collected. The mountain of evidence would conclusively pinpoint Toms as the culprit. Despite Tom's best efforts to disguise the clues, modern forensic science could detect traces of evidence beyond what is visible to the naked eye. Today, using tools that were unimaginable 2 centuries ago, scientific proof would convict Toms, beyond a shadow of a doubt. How much more evidence would modern investigators have about Toms than investigators from 200 years ago?
In racing, the fastest time wins. But for these athletes, it's hard to separate time from distance.
1 second to me means 14 meters, because in my event, that's what it translates to.
Mike McCarthy knows how important time is. He once lost a world championship by 0.04 of a second. Here at the Olympic Training Center, the focus is on saving fractions, 100th, even 1,000th of a second. In the past, these competitions were judged using handheld stopwatches, but this created errors in the measurements. Judges used only their eyes, and by the time they reacted to what they had seen and stopped their watches, they added an error of a fraction of a second. Now, these events are judged using automatic timers and photo finishes. There is no longer a need to worry about human error. In many sports, time is of the essence. Keeping time accurately is the name of the game. A photo finish shows that 0.02 of a second separates the riders in this race. If needed, this race could be judged to 100,000th of a second or better. Technology allows for such accurate measurements.
That's about an inch at the speeds that the riders are going these days.
And even a gust of wind in the wrong direction can rob a competitor of a few thousandths of a second. With this degree of accuracy with measurement devices, the national bicycling team has developed strategies for beating the clock. How do cyclists shave off thousandths of a second? Everything has to be taken into consideration. The cyclists must go from a standing start to being able to get their wheels turning with incredible speed. Every component has been streamlined to reduce wind resistance. The pedals are smaller for better cornering, and the front wheel has been made ultra small, which allows for better drafting. The smaller the front wheel, the shorter the whole length of the bike.
We're saving just centimeters. Closer you can ride to the guy in front of you, the more energy you can save. But then, of course, it is more dangerous. You're a centimeter away from a crash the whole race. Well, we just started using this helmet this year. It's basically just a fairing for the head. It's very narrow on the front, which makes it more aerodynamic. We have actually golf ball divots on the top, which smooth the airflow out and help you to go faster yet. And then it's shaped long to fit behind the bridge of the neck to help smooth the airflow out as well.
When you go to the line on one of those bikes, you really feel like this is a huge advantage for me. That when the exercise physiologist and the biomechanist come to you and they say, this bike is worth 2 seconds on the line, and you know that the guy on the other side of the track is 1.5 seconds faster than you, you might have a good chance to beat him.
Mike knows what it's like to have time on his side. After his close second-place finish at the World Championships, he came back the next year to win it. Why is it important to be able to measure time accurately in a race? As you watch this segment, think about how the ability to make accurate measurements has changed navigation. The aircraft carrier George Washington is one of the most sophisticated ships afloat. To find their way in the ocean, these sailors have every conceivable high-tech navigation instrument. So what in the world are these sailors doing still using the stars to navigate? And why is the key piece of equipment an accurate clock? This centuries-old technology still has relevance today. And here's why. You can use just the stars to determine your latitude, your position north or south. But to determine your longitude, your position east or west, you also need to know the difference in time between where you are and the time at a fixed location. And for that, you need an accurate clock. Having the wrong time could result in being off course by hundreds of kilometers east or west. This was the situation that sailors were in centuries ago. When scientists speak of the accuracy of a measurement, They mean how close the measurement comes to the real value. Using star charts allowed sailors to accurately measure the local time. Precision, on the other hand, is how close a group of measurements are to each other. Up until a couple of hundred years ago, there was no way to keep a clock accurate on board a ship. And using the sun and stars to tell time was not accurate enough to determine exact longitude. Astronomers at the Royal Observatory in Greenwich, England, offered a solution to one part of the problem. They published an almanac showing the positions of the stars over Greenwich any day of the year, any time of the day. Sailors used a tool called a sextant to measure the distance of a star from the horizon. By comparing their findings to the charts in the almanac, sailors could determine the local time much more accurately. Greenwich also became the fixed location for time measurement. But sailors still needed a way to keep track of that time.
We have predetermined stars that we've gone and figured out for to shoot, which are the heavy bodies. For example, they shoot code shaft. Okay? We take the star down to the horizon. The most important thing about using celestial bodies, be it the sun, the stars, the moon, or the planets, is time. Everything is related to time.
By using new technologies, the sailors on the George Washington can measure the local time and compare it to the time in Greenwich. But centuries ago, the only clocks that existed were pendulum clocks. They couldn't be used on moving ships because the ship's movement on the ocean disrupted the pendulum's regular movement. A race began for a mechanical clock, or a chronometer, a device that would be able to keep accurate time on the open sea. Jim Michaels, a modern-day watchmaker, explains the history of this device.
There was a commission set up in England that would offer a prize of £20,000, I believe it was, to anyone who could come up with a chronometer that would meet very strict standards.
The year was 1714, and the man who stepped up to the challenge was a carpenter named John Harrison. He spent 40 years developing this hairspring mechanism that replaced pendulums. During a final test voyage to the Caribbean, it lost only 7 seconds. Harrison won the £20,000, which translates to $12 million in today's money. A worthwhile expenditure, according to the navigator of the $3 billion George Washington. The invention of the chronometer was a major step in the technological advancements that helped the navigator of the George Washington do his job.
What we're doing right now is setting ourselves into the wind, which we are. We're coming up on speed so we can get the minimum winds over the deck for the heaviest aircraft, which in this case is going to be an A6 tanker.
The carrier must always be moving, into the wind for takeoffs and downwind for landings. With all this turning and maneuvering, it would be easy to get off course. Although the first line of action is to use satellites and the Global Positioning System, or GPS, sextants provide a reliable backup.
If something does go wrong with the electronics, if a satellite fails for one reason or another, I have my quartermasters that can still shoot the stars and still give me a good fix. And as you can see, where he plotted our fix today was within 1,000 yards or less of where the GPS had us, which is extremely accurate in open ocean.
Satellites can fail, but the stars remain a constant. That is why every day on this high-tech vessel, the quartermasters still shoot the stars. They still check the ship's clocks, and they report their status to the captain.
Sir Deck sends his respects and reports the approaching hour of 12 o'clock. All chronometers have been compared. Request permission to strike 8 bells on time.
Strike 8 bells on time.
What 2 readings are necessary to determine longitude?
When astronaut Neil Armstrong set foot on the moon, he made history. Tucked inside Armstrong's pocket was a memento from what many consider the most remarkable day of the 20th century, the day humans first flew in a motorized, controllable aircraft. December 17, 1903, Kitty Hawk, North Carolina. Wilbur and Orville Wright's Flyer travels 36.5 meters in 12 seconds. This one flight will change the world.
500 years from now, when people are looking back and to say, well, what was special about the 20th century? It's the century where humans began to fly.
Nearly 100 years after the Wright brothers' historic flight, a hand-picked team of aviators, engineers, and historians assembles in a Virginia workshop. Their mission to exactly recreate the Wright brothers' airplane and fly it 100 years to the minute from the first flight. It's a difficult job because the Wrights left behind no blueprints, and the original plane is long gone, though pieces do remain. The engineers must become investigators, extracting information from airplane parts, photographs, and letters. This process is called reverse engineering, and it involves analyzing an object, or in this case, parts or photographs of an object, in order to duplicate it. The team will spend 4 years creating an exact replica of the Wright Flyer. They'll use the same tools the Wrights did, then test their progress with tools the Wrights never dreamed of: computer models, cutting-edge laboratories, and a NASA wind tunnel. Along the way, they'll discover the step-by-step design process the Wrights undertook. What is reverse engineering? As you watch the following segment, think about the design process the Wright brothers followed as they invented the airplane. In rural Virginia, a team of people labors to design an exact replica of Wilbur and Orville Wright's first airplane. Among them are historians who piece together the Wright brothers' story and their place in our nation's history. In 1900, Wilbur and Orville Wright are the owners of a successful bicycle shop in Dayton, Ohio. Both are lifelong tinkerers, and their inventive minds are focused on the skies.
Flight, in fact, was the overriding problem at the turn of the century. If you were looking around for a challenge, especially if you were interested in science and technology, if you were interested in that kind of problem solving, it was the big challenge.
The Wright brothers decide to take on the challenge of motorized flight. People all over the world are trying to fly. Getting into the air isn't the problem. Staying there is. Wilbur decides to write to the Smithsonian Institution requesting any papers the museum has about the subject of flight. He reads everything he can get his hands on, and concludes that previous attempts at flight failed because the pilots couldn't control the aircraft.
And being bicycle riders, they knew they had to be able to control the airplane at all times. They didn't care whether it was stable or automatic in any manner, shape, or form. They just wanted it to be controllable.
The challenge is controlling the airplane in all 3 dimensions: side to side, or roll, up and down, or pitch, and left and right, or yaw. How could the Wrights achieve better control? The solution comes in the most unexpected way.
One day, Wilbur was in the bicycle shop. The Wright brothers were bicycle manufacturers, and he was talking to a customer who he had just sold an inner tube tire to, and he was kind of fiddling with the paper box that the inner tube came in.
Wilbur has a flash of inspiration. He envisions a set of airplane wings modeled after the box.
This would be the upper wing surface of a biplane, and this would be the lower wing of the surface of a biplane.
Wilbur theorizes that he could twist the wings and control the direction in which the airplane moves. It's called wing warping. To test his theory, Wilbur builds a kite inspired by the cardboard box.
What he had was two strings with levers that attached to the front end of the kite, and he could actually, with sticks, actually wing warp the airplane from the ground. And story goes that it would dart towards the ground and then pull up, and so he had the control that he wanted in the process.
The next step, build a larger version of the kite, a glider big enough for a pilot to fly. In early 1900, Wilbur builds a full-scale glider in the back of the Wrights' bicycle shop. A glider has no motor and relies on the power of the wind to stay aloft. Fortunately for the team retracing the Wright brothers' steps, both Wilbur and Orville had a keen interest in photography. They documented nearly everything they did.
We couldn't do what we're doing today, the reverse engineering of the process, if we didn't have those photographs to analyze.
Using the photos, the team builds a 3D computer model of the first Wright glider.
One of our pieces of information is actually a photograph taken of a wreck. But even with the aircraft in that condition, there's a wealth of information in here for us.
In the fall of 1900, Wilbur travels from Ohio to Kitty Hawk, where the winds are strong and frequent. The brothers tow the glider to Kill Devil Hills for a trial run. From a running start, Wilbur lifts off the sand and shoots up 6 meters. Wilbur is able to control the glider, thereby proving his theory of wing warping. But there's bad news, too. The aircraft doesn't have enough lift. A successful plane will have to carry a motor weighing about 90 kilograms. It's back to Dayton and the drawing board. In the late fall of 1900, the Wright brothers begin what will be a 3-year process of designing, testing, and redesigning their aircraft. Their second glider performs badly at Kitty Hawk. It still doesn't produce the kind of lift the mathematical formulas say it will. Wilbur and Orville seriously consider giving up. Instead, they conclude the formulas are wrong. They build a wind tunnel and test 200 miniature wings of all shapes and kinds. They observe how the different wings perform. In their homemade wind tunnel, the Wright brothers invent the modern wing, and something more.
The Wrights established kind of a systematic method to their approach, and they would solve a set of problems, use their solutions and their findings from their testing and their flight trials, and go back to the drawing board and come up with a new design. And there was a real step-by-step evolutionary approach to it, what we would call a scientific method, that nobody else really seemed to grasp onto at the time.
Using that method, the Wright brothers revolutionize rudder design, invent the modern aircraft propeller, and direct the design of a powerful but lightweight engine. In the spring of 1903, Wilbur and Orville begin building their aircraft. It's so big it won't fit in their workshop, so it has to be built in parts. The brothers dub it the Whopper flying machine. As they assemble the new flyer, the Wrights practice on their old glider. 100 years later, the pilots on the recreation team do the same. They train in a full-scale flight simulator and in a replica of the Wright glider.
It is a little bit squirrely. It's very, very light. It reminded me of flying a Kleenex. It's just so light and so airy and almost has a mind of its own, but it's very capable of being controlled.
December 17th, 1903, launch day. It is cold and cloudy with wind gusts up to 48 kilometers per hour. The conditions are not favorable, but Wilbur and Orville are determined to fly. At 10:35 a.m., Orville is in the plane. He releases the line holding the motorized flyer. It wobbles ahead into a 43 kilometer per hour wind. Wilbur runs alongside to steady it. 2/3 down the track, Orville feels the plane lift and yanks the controls. 12 seconds and 36.5 meters later, Orville lands in a new era. The day after their historic flight, Wilbur and Orville telegraph the good news to their family, along with the line, "Inform Press." Incredibly, only 3 newspapers pick up the story of the century.
Process of invention was over. They had a product for sale now, and that became their new problem. How do they sell this thing, this product of theirs?
For 6 years, there are no buyers. Then, in July of 1909, the Wright brothers auditioned for the U.S. government. Their challenge, to stay in the air for 1 hour. 73 minutes later, Orville lands. The skeptics are silenced, and the inventors are in business. December 17th, 2003. 40,000 people gather to witness the reenactment of the Wright flight. There's pouring rain and very little wind. 100 years to the minute from the Wright brothers' flight, weather forces a delay. Within hours, though, the team is ready and the replica starts down the launch track. There is just not enough wind for it to take off. The recreation team is disappointed, but philosophical.
I was proud of everybody. We got the airplane ready. We got here. Some things got left undone, but bottom line is we were ready.
If anything, the failure just highlights the genius of the Wright brothers. It demonstrated just how hard it was for Wilbur and Orville to build an airplane from scratch and actually fly it. And the failure contains a valuable lesson about the design process. There are often failures on the road to success. What are the 6 steps in the design process?
This is Robert Hooke. It's 1665 and microscopes have only just been invented. For the first time, scientists were able to look at things too small to be seen by the naked eye, and Robert Hooke was fascinated by what he saw. Oh, this piece of cork seems to be made up of tiny compartments, like lots of little rooms all fitted together. Rooms or cells. Yes, they look like cells. Hooke became famous for being the first person to use the word cell to describe the room-like structures inside his piece of cork. He spent many hours carefully drawing their honeycomb pattern. In 1833, one of the leading botanists of the time was Scotsman Robert Brown. Having travelled the world and collected hundreds of plants, he made a startling discovery. There are so many plants, all of them so different, except when I look at them close up. Every plant is made up of cells, and inside every cell there is a sort of dark, dense blob. Whichever plant I choose, it's always the same. I can find not one single exception. Robert Brown called the blob-like structure inside each cell the nucleus. About this time, scientists were beginning to realise that all living things were made of cells. A few years later, in 1839, 2 German scientists, Dr. Schwann and Dr. Schleiden, came up with a theory which was to change the way scientists viewed the living world. Dr. Schwann? Yeah, Dr. Schleiden. That's them. I think we have discovered something completely amazing. The material inside the cell moves about. Very slowly, I grant you, but still, she moves. We can only conclude one thing from this. You mean? Yeah, the cells are alive. Oh. This got me thinking. Sometimes a single cell exists on its own. But more complex creatures like you or I are made up of millions of living cells, all working together, All living things are made of cells, no matter how big or small, like a plant. Even this fly. Yeah. And the theory they eventually came up with was that cells are the basic building blocks of life.
Which early scientists contributed to the development of the cell theory? As you watch this segment, think about specialized cells and their functions. Everything that is alive, from the smallest organism to human beings, is made of cells. Even though we can't see them, trillions of cells within our bodies work together to help us do what we need to do. Human cells come in all shapes and sizes, and they carry out specific functions. Skin is made of cells, and so are our hearts, our muscles, and even our brains, where cells send electrical signals from one to another so that we can think. Throughout our bodies, cells are constantly dying and being replaced by new cells. New cells are produced through mitosis, the process in which one cell divides into two identical cells. Since cells are being replaced on a regular basis, most cells in our body are actually younger than we are. Our cells work together in extraordinary ways. The heart, for example, is made up of muscle cells with the characteristic of contracting. Separately, heart cells pulse to their own rhythm, but together they beat as one. Millions of heart cells work together to give us a heartbeat. The surface of the trachea, the tube that leads from the throat to the lungs, is moist with mucus. Bacteria and dust stick to it. The cells on these surfaces have tiny microscopic hairs called cilia that stop foreign substances from entering your lungs. These cilia wave back and forth and push the mucus up to your throat. You might have heard the saying, blood is thicker than water. Well, the saying is true. That's because our blood is packed with millions of cells that float in a river of plasma. Most of these are red blood cells. They're one of the few types of cells without a nucleus. These flexible discs can bend and squeeze through the smallest capillaries in our body. They get their red color from hemoglobin, a protein that carries oxygen. When blood passes through the lungs, millions of red blood cells pick up oxygen and carry it to the rest of the cells in our body. Our blood also holds an army of white blood cells that function in fighting invaders. They defend our bodies against illness and disease. When foreign microorganisms like viruses or bacteria enter the body, white blood cells can recognize them and rush to destroy them. Other blood cell fragments, called platelets, protect our bodies by controlling bleeding. When we cut ourselves, platelets stick to each other and form blood clots that stop the bleeding. Threads of protein form a tight mesh, trapping blood cells and keeping out harmful germs. This becomes the scab. Eventually, the scab falls off, leaving behind a strip of new skin. Although you may think of your skin as being alive, the outermost layer of skin, called the epidermis, is actually a layer of dead cells, about 25 cells deep. That's about half as thick as a sheet of paper. Over time, these surface cells flake away to be replaced by new cells that form underneath. It takes about 3 weeks for new skin cells like these to form and take the place of the older cells. This is what dead skin cells look like under a microscope. We shed billions of these cells every day, about 22 kilograms in a lifetime. Believe it or not, a lot of the dust in our houses is actually dead skin cells. This constant renewal of cells keeps our skin healthy. Just as animals are made of cells, so are plants. But plant cells are a bit different from animal cells. A typical animal cell is like a tiny blob of jelly. Take a look inside, and there's a nucleus surrounded by a jelly-like substance called cytoplasm. The outer covering is called the cell membrane. Typical plant cells have a box-like shape. Like animal cells, they have a nucleus, cytoplasm, and a cell membrane.
But plant cells also have a cell wall. It's a rigid outer covering made of a strong material called cellulose. Inside most plant cells are green organelles called chloroplast. They absorb the sun's energy and carry out photosynthesis, a chemical process during which water and carbon dioxide are made into sugars. The leaf cells of most plants have many chloroplasts, which contain the green pigment chlorophyll, which gives leaves their color. Root cells, on the other hand, are usually colorless. That's because roots normally grow underground. The structure of root cells is well suited to taking in water. These cells have hair-like extensions that increase their surface area, thus increasing their ability to absorb water. Just as in the human body, cells in different parts of the plant do different jobs. What is the function of root cells in plants?
Here at the National Cancer Institute in Bethesda, Maryland, Tina McDaniel is waiting for a miracle. The 17-year-old high school senior has been living with a deadly form of cancer called melanoma. Melanoma is a skin cancer often, but not always, caused by exposure to ultraviolet radiation from the sun. Humans have a defense against the harmful effects of sunlight. A chemical pigment called melanin absorbs ultraviolet radiation and keeps it from damaging the body's tissues. These are melanin-producing cells. These cells lie just under the top layer of your skin, but it is likely that these cells are different from yours. Exposure to ultraviolet radiation over time has changed the way these cells usually behave. Instead of growing, reproducing, and resting afterwards like most cells, these cells continue to grow and reproduce without a rest period. The result is a mass of abnormal cells called a tumor, which may interfere with the functions of other cells around it. These cancerous cells can also spread throughout the body. That's what's happening to Tina. But there is hope. Dr. Steven Rosenberg of the National Cancer Institute is Tina's physician. The challenge for him and other physicians is to stop the uncontrolled growth of cancer cells. Dr. Rosenberg is working on an experimental new treatment that uses the body's natural immune response to fight the melanoma. The treatment has given many of Dr. Rosenberg's patients a chance to overcome the disease. It may do the same for Tina. What do doctors believe is the primary cause of melanoma? As you watch this segment, think about how normal cells grow and divide and how cancer cells behave. Cancer is the general name for a disease characterized by the abnormal and uncontrolled growth of cells. Often, cancer cells accumulate in one area of the body and form a growth called a tumor, like some of the dark spots on this woman's skin. Normally, cells go through a period of growth and division called the cell cycle, which is divided into 3 stages: interphase, mitosis, and cytokinesis. During interphase, the cell grows to twice its normal size and copies of the cell organelles are made. Also during interphase, the cell makes copies of its DNA through a process called DNA replication. As a result of replication, each new daughter cell will contain an exact copy of the genetic information from the parent cell. The second stage of the cell cycle is mitosis. During mitosis, a copy of replicated DNA is distributed to the daughter cells, and a new nuclear membrane forms around each region containing DNA. Cytokinesis is the final stage of the cell cycle. During cytokinesis, the cell membrane pinches off, forming 2 new cells. Each new cell contains half of the cytoplasm, roughly half of the duplicated organelles, and an exact copy of the parent cell's DNA in each nucleus. Doctors in hospitals around the world are studying the differences between normal cells and cancer cells. One way that they do this is by examining human skin. Using a scalpel, doctors can scrape skin cells off pieces of skin and into a solution that will preserve them. These cells can be seen under a microscope that makes them look 300 times larger than they really are. On the left side, you can see time-lapse photography of healthy skin cells growing normally. On the right side, you can see cancer cells. The normal cells on the left will ultimately stop dividing. The cancer cells on the right will not stop dividing. As cancer tissues grow and tumors form, they prevent nutrients from getting to healthy cells, which damages and kills the healthy cells. Cancer cells can also travel to other parts of the body through the bloodstream or the lymphatic system. As the abnormal cells travel through the body, they can become lodged in many of the body's tissues where they continue their abnormal growth, and thus spread the disease throughout the body. Though research continues, scientists are not certain what causes cancer cells to grow uncontrollably. Doctors have used surgery, chemotherapy drugs, and radiation treatments to fight cancer. But today, scientists on the front lines of cancer research are exploring various ways of treating cancer, including the possibility of using the body's natural defenses to rid the body of abnormal cells.
And when we use surgery or radiation therapy or chemotherapy, we're destroying normal cells as well as cancer cells because they can't distinguish between the two. But the immune system can. It can distinguish a single molecule that's different on a cancer cell than a normal cell.
Using an experimental new cancer vaccine made from the amino acids found on a cancer cell's surface and a hormone called interleukin-2, Dr. Rosenberg has been able to destroy cancer cells in melanoma. The treatment is simple. First, the patient receives an injection of the vaccine. The vaccine enables a specialized white blood cell to identify the cancer cells and attack them.
These are the immune white blood cells, the lymphocytes, that we're trying to activate by our vaccines. And as you can see in these remarkable images that are actually taking place in culture, the lymphocytes attacking the tumor. And as it attacks and attaches to the tumor, we see these projections in the tumor cell which indicate that it's being destroyed, its membrane is being dissolved.
Dr. Rosenberg's new treatment has given hope to many people suffering from melanoma. Some scientists hope that his technique may work in the fight against other forms of cancer. Scientific breakthroughs don't happen every day, but each new treatment gives both doctors and patients faith that one day we will beat cancer for good. How does normal cell growth differ from cancerous cell growth?
This is Infinity Duncan. At the age of 4, she has already experienced many health problems that typically strike adults. Infinity suffers from sickle cell disease, a genetic disorder that interferes with the normal synthesis of the blood protein hemoglobin. People with sickle cell disease produce a defective form of hemoglobin, and that results in painful symptoms. This defective protein results in red blood cells that are rigid instead of flexible, and sickle-shaped instead of disc-shaped. These rigid blood cells do not easily flow through the body's tiniest blood vessels, which blocks the delivery of oxygen to some cells and also causes pain. Once a week, Infinity comes to Fairview Hospital in Minneapolis for treatment. Regular blood transfusions lessen her chances of experiencing an acute episode of the disease known as a sickle cell pain crisis. That's because the new blood contains red blood cells with normal hemoglobin. Blood transfusions are helping Infinity live a normal life. Unfortunately, regular blood transfusions can cause iron to build up in the body. This buildup of iron can damage many of the body's organs. To reduce this iron buildup, the doctors must give Infinity specialized medication. Having sickle cell disease hasn't stopped Infinity from doing many of the things that young people her age do. But that doesn't mean that her doctors aren't searching for a cure. For the past 5 years, Dr. Lakshamnan Krishnamurti and his colleagues have been trying to find a cure for Infinity and the millions of other people who suffer from sickle cell disease. Because sickle cell disease is a genetic disorder, there is a possibility that new research in genetics may give sickle cell sufferers a chance at a healthy life. What protein is synthesized incorrectly in sickle cell disease? As you watch this segment, think about how a mutation in DNA can result in a defective protein. Everyone on Earth is different. Some people are tall, while others are short. Some have dark hair and dark skin. Others have light hair. Some have blue eyes, while others have brown eyes. These phenotypes, or visible traits, and many other non-visible characteristics are the result of proteins that are synthesized in our body's cells. Our cells can manufacture a total of more than 100,000 different proteins. These proteins are made during a process called protein synthesis. Most of the time, protein synthesis makes the right protein, but sometimes things go wrong. To understand how this happens, it's necessary to look at just how protein synthesis works. The instructions for synthesizing proteins come from inside the nucleus of our cells, from the DNA molecules that make up our chromosomes. Specific segments of DNA carry instructions for making each protein. These segments are called genes. DNA is made up of 4 kinds of nitrogen bases: adenine, thymine, guanine, and cytosine. These bases are identified by the initials A, T, G, and C. A complete DNA molecule has a structure similar to a ladder. The rungs of this ladder are made up of pairs of nitrogen bases. Each nitrogen base only pairs up with a certain other nitrogen base. For example, A only pairs with T, and G only links to C. A single gene on a chromosome may contain anywhere from several hundred to 1,000,000 of these bases. The instructions or code for making a protein is the sequence or order of the nitrogen bases. For example, the sequence CCTGAA is a code that might result in one protein, while CCTGTA might make a different protein. Within the cytoplasm of the cell, an organelle called ribosome is the place where proteins are synthesized. But sometimes one base is substituted for another while inside of the ribosome. Though it is rare, this substitution can lead to big changes in the body. These substitutions or changes are called mutations. Sickle cell disease is an example of an error in the DNA code resulting from a mutation of one nitrogen base. In only one place, a T replaces an A. It doesn't seem like much of a difference, but in this case, that one different base leads ultimately to a protein with one amino acid that is not normal. That one difference is enough to cause a change in the structure of hemoglobin, which causes red blood cells to sickle. Blood cells containing this defective hemoglobin cannot easily pass through the body's tiniest blood vessels. The vessels become clogged, and oxygen is cut off from the body, causing permanent damage to tissues. This blockage also causes intense pain. Known as a sickle cell pain crisis, this intense pain strikes both adults and children who suffer from sickle cell disease. This genetic disease affects more than 5 million people around the world. Doctors have been searching for an effective way to treat sickle cell disease since the early 1950s. Though there are medications and therapies that can help people who suffer from sickle cell disease, these treatments do not work for everyone. But researchers working with the Human Genome Project, a program designed to find out the genetic causes of diseases, think that sickle cell disease may be targeted as one of the first diseases to benefit from gene replacement therapy. Scientists at the Lawrence Livermore Laboratories in Berkeley, California are conducting experiments with mice. Research has shown that injecting a special kind of sickle cell gene into the bone marrow of these mice can help them produce healthy round blood cells. And there is a possibility that this treatment may one day work for people with sickle cell disease. Where do the instructions or code for protein synthesis originate?
This is Tasmania. Located just south of Australia, Tasmania is a land of craggy mountains and mysterious forests. Like the rest of Australia, Tasmania has kangaroos, wombats, and many other marsupials that carry their young in pouches. But there is one species of marsupial that's no longer found here: the thylacine. For millions of years, thylacines were the island's chief predators. Since these fearsome creatures with dog-like heads and striped backs are believed to be extinct, computer animation is the only way that we can see the Tasmanian wolf, also known as the Tasmanian tiger, in its natural habitat. Today, only the tiger's ghosts remain on the Australian continent. The last known thylacine died more than 60 years ago. The species is now thought to be extinct. The destruction of the Tasmanian tiger was deliberate. In the 1800s, the government offered rewards for killing the animals because settlers feared they would eat their sheep. No one thought the thylacine would be missed. About 1,000 kilometers north of Tasmania is Sydney, Australia. One of the city's oldest institutions is the Australian Museum. Its director is Professor Mike Archer. He admits he's obsessed with thylacines and their fate, which is why Professor Archer and other scientists at the museum have set out on a bold experiment to try to reverse extinction. If he can extract DNA from this thylacine specimen, maybe one day scientists can create a living thylacine clone.
Okay, now this is going to be the tricky thing.
A clone has the same DNA as the organism from which it was produced. If Professor Archer can clone the thylacine, he will have done something no one has ever done before: brought an extinct species back to life. Explain what Professor Archer wants to do. As you watch this segment, think about how cloning may be used to bring back extinct species. This thylacine, or Tasmanian tiger specimen, has been kept in alcohol for almost 140 years. The alcohol has helped to preserve the animal's DNA, the genetic material that contains the building blocks of the organism.
The first bits of a miracle are in your hand.
It seems to be just a tiny amount of sort of cotton wool ball down there, very small one.
Fantastic.
After 140 years in alcohol, some DNA has survived. This is the first step toward cloning the Tasmanian tiger. Professor Archer explains the significance of the existing DNA.
My heart is beating really fast. I mean, we have dreamed about this thing for years. I thought about it, at least 20 years ago, and I never really thought we'd get to this point. I mean, it was impossible, told by many people to us we'd never get here. The dream is starting to become a reality. I mean, we're not there yet, but step one, the fantastic step of actually getting DNA, is occurring. So I could not be a happier man than any in the whole world. We are really going here.
When most of us think of cloning, we think of the sheep Dolly, the first mammal cloned from an adult sheep cell. But since then, scientists have also cloned other animals through a process called nuclear transfer. In nuclear transfer, scientists take a cell from the animal to be cloned and remove the nucleus, the place where the DNA is stored. The nucleus is then inserted into an egg cell from a host animal that has had its own nucleus removed. The egg is zapped with an electrical charge to fuse the new nucleus in place. If the cell begins to divide properly, an embryo is created that can be implanted in the womb of a host mother. The Australian scientists' success in extracting DNA from the thylacine specimen was a moment Mike Archer couldn't wait to share with the world.
Ladies and gentlemen, we are here to announce what is probably the biological equivalent of human beings taking the first step on the moon. Nobody has ever done what we have just done. It is the threshold of what will be a very exciting series of opportunities to maybe question whether extinction really is forever.
But despite the extravagant claims, science is still a long way from being able to clone a thylacine, as Mike Archer knows only too well. The DNA removed from the specimen has broken down over time. Normally, long strands of DNA provide codes that determine the characteristics of an organism. But the thylacine DNA strands are broken. Scientists will have to guess how to put them back together. This will make it very difficult to recreate the original thylacine DNA code. When scientists cloned the sheep Dolly, they didn't have to worry about that because they were able to extract the DNA completely intact. Archer and his colleagues don't have that luxury. Their task is similar to trying to put together a jigsaw puzzle with millions of pieces, and many of the pieces are missing.
How do we put it together as chromosomes? How do we know how many chromosomes we should have? How do we get the DNA viable? It's pickled.
Right now, the scientists are trying to learn as much as possible about the Tasmanian tiger DNA they do have. And while the world is still a long way from cloning an extinct animal, the work of Mike Archer and other scientists brings us a little closer each day to making what was genetically impossible possible. And who knows, maybe one day the thylacine will roam the hills of Tasmania again.
I think we can do this. We may be asking too much at the moment of technology. We may be ahead of what technology can actually do at this point. But if the vision stays out in front of the game and leads, I think we've got that chance. We've got that chance of having our dream actually come true.
Why do scientists want to clone the Tasmanian tiger?
In the mid-1830s, biologist Charles Darwin made a dramatic scientific exploration of the Galapagos Islands. Darwin said the Galapagos seem a perennial source of new things. And just as in Darwin's day, recent expeditions to Galapagos continue to discover new species and new information on how they may have evolved. Today, most new Galapagos species are found in the deep waters around the islands. Modern technology and research equipment now allow scientists to explore regions that Darwin could only dream of. And the findings of modern scientists fill them with as much wonder as Darwin experienced during his famous voyage.
As we approach this, you don't know what it's going to be. You come in close and begin to get a little more analytical and a little less emotional. But there's no getting away from the thrill that comes with realizing that what you come upon is something new and perhaps something that's never been seen before by anyone.
We think that this is probably a new species, one that's not been described before, and I'm certain that this is the only footage available of cirrate octopuses in good condition like this. This is an extraordinary deep-sea fish. Common name is the tubai. This is probably the first living specimen anybody has ever had in a position to be able to photograph. Pelagothuria is completely unique among the sea cucumbers in that it never spends any time on the sea floor. This is the first time that anybody has had the opportunity to see what it feeds on and to be able to characterize its role in the deep sea community here.
This is the sort of thing that you hope for on an expedition like this. You know you can't count on it, but this is what you're hoping you're going to find. Something new, something undescribed, something that'll help you put together the little bit of the unknown that you have a chance to reassemble in your mind.
In what habitat are most new Galapagos species being discovered? As you watch this segment, Think about how the different species of the Galapagos Islands provide evidence to support the theory of evolution. The 13 large Galapagos Islands formed 3 to 5 million years ago when undersea volcanoes created an island chain 960 kilometers from any other land. The organisms that flew, floated, or otherwise made their way to these islands were isolated. Over millions of years, these organisms adapted to their unique island environment differently than the species back on the mainland. In 1831, 22-year-old biologist Charles Darwin and the crew of the HMS Beagle set sail from England on what would become a 5-year expedition around the world. When they reached the Galapagos in 1835, the unique creatures they found there fueled Darwin's scientific curiosity. Darwin found many species he had never seen before. He also noticed differences within the Galapagos species from one island to the next, such as tortoises with different types of shells and finches with different types of beaks. Darwin made detailed notes and sketches of the species he encountered and collected thousands of specimens. When he returned to England, he spent the next 20 years analyzing and interpreting his findings and doing further research in biology and geology. Finally, in 1859, he presented his groundbreaking hypothesis. In The Origin of Species by Means of Natural Selection, Darwin put forth a controversial argument for his day that all species changed over time, or evolved. Darwin argued that species change over time and, as a result, become adapted to their environments. For example, the blue feet of this booby are an adaptation. Females are attracted to males with bright blue feet, so the males with this adaptation are more likely to reproduce and pass this trait on to their offspring. Likewise, if an adaptation gives an organism a competitive advantage over others of its kind, such as strong legs for swimming and diving here on Galapagos, that organism is more likely to survive and pass the trait on to the next generation. Over time, these beneficial adaptations can build up in isolated populations, like those of the Galapagos, and may give rise to a new species. Galapagos's marine iguana provides compelling evidence for Darwin's ideas. It's believed that the ancestors of marine iguanas were originally land iguanas that may have come to the islands on floating logs. But food on these volcanic islands was scarce. During the competition for food, some iguanas found success by venturing into the waters. The land iguanas with slightly better tails for swimming or shorter snouts for eating off of rocks had an advantage over iguanas without these traits. Darwin argued that over millions of years, the iguanas with physical traits that made them good divers had a better chance of surviving and therefore of producing more offspring. Over the generations, a greater percentage of iguanas inherited these beneficial traits. Eventually, the marine iguanas evolved into a separate species from the land iguanas. Darwin called this process natural selection. Organisms with beneficial adaptations were effectively selected by nature to have a better chance of either surviving in their current habitat or occupying a new habitat. Darwin found creatures with unique adaptations all across the Galapagos, and modern researchers continue to find them. For instance, the swallow-tailed gull's big red eyes give them a competitive advantage over other gulls in finding food.
The swallow-tailed gull is unusual among gulls in that it has a very much enlarged eye, which is a beautiful adaptation for feeding nocturnally. The swallow-tailed gull feeds mainly on bioluminescent type organisms that light up the surface of the sea at night.
So these gulls have evolved to take advantage of a food source that no other gulls can. These adaptations help to ensure their survival. There is a different species of finch on each Galapagos island. Isolated on each island, each has its own unique behaviors and beak size and shape. Some eat like woodpeckers, some eat cactuses, and others use tools, but they each find food in their own way and pass their successful adaptation on to the next generation. Beneficial adaptations can also help species live side by side and still avoid competition. For instance, hawkfish with long snouts can coexist in the same waters as the short-nosed variety because one finds food in crevices while the other finds it on top of rocks and coral. A spectacular and seemingly negative adaptation can be found in many of the other marine creatures here. Their bright coloration would appear to make it easier for predators to see them.
And it's hard for us to understand as light viewing human beings at the surface of this planet why these fish are so darn colorful. And with the sub, with the lights on, we're just amazed, kind of whacking ourselves in the head saying, why the heck is that fish bright red, yellow, orange, what have you? And we turn the sub lights off and allow our eyes to adapt to that very, very low light level, even 1,200 feet below the surface. And you realize that there is some light penetrating through this clear tropical water. And the fishes that are just jet black, which stand out like sore thumbs, but these very colorful fishes all of a sudden fade right into the habitat. They look like the sea fans. They look like the broken rock reef. That color is very adaptive. And why it is so beautiful is beyond me. I can't imagine there's an advantage to that.
So, a trait that would be harmful for a shallow water fish is actually beneficial here in the deep waters of the Galapagos and has become widespread in the marine population. Over 170 years ago, the amazing creatures of the Galapagos made Charles Darwin question where species come from, and they are still providing answers today.
What advantage do larger eyes give to the Galapagos swallow-tailed gull?